Assalamualaikum and hi, my name is Atisha binti Afandi. Me and my teammates, which is Siti Aisyah Rosli and Nur Izzati binti Nordin, will be doing a video assignment together, and I will be the first presenter. Me and my teammate choose topic gene recombination, which is on chapter 6. There is transformation, transduction, and conjugation to generate variation in prokaryotes. The meaning of transformation is a bacterium take up a foreign DNA that floating in its environment. For transduction, movement of genes from one bacterium to another by a virus. For conjugation, the process where DNA is transferred between bacteria through a tube between cells. I will explain about transformation. Transformation is a change in an organism characteristic because of the transfer genetic information, meaning that bacteria will be pick up the suitable cell and will get into the genetic composition. But if it's not suitable, it will break down by the enzymes. After that, we will get a new bacteria with a new genetic composition.
Transformation occurs when bacteria take up extracellular DNA and incorporate it into their genomes. Typically, this occurs when one bacteria lyses, or splits open, releasing its genetic contents, and then another bacteria comes by and acquires it. Bacteria able to bind large amounts of DNA are termed competent. Competency is a state of increased cell wall and cell membrane permeability that allows cells to uptake DNA. Many bacteria are naturally competent, and so actively bind environmental DNA.
That is all from me. Next presenter will be Siti Aisyah Rosli, and she will be explain about transduction.
Hello and Assalamualaikum everyone. So today I will be talking about transduction. In general, transduction is a movement of genes between bacteria by bacteriophage, which also meant by bacteria eater or virus that infect bacteria called phage. There is two types of transduction. Firstly, generalized transduction, which involves in lytic phase. The bacteriophages only carries the pure bacterial DNA, not the phage DNA, and this pure bacterial gene is inserted into the recipient cell. And secondly, the specialized transduction involved in lysogenic phase, and it contains both the bacterial DNA and the phage DNA. For generalized transduction, firstly, the bacteriophage attacks the bacterial cell. The bacteriophage injects its genetic material, which then penetrates the bacterial chromosome. The phage DNA then controls the whole cell machinery. This causes the degradation of the bacterial chromosomal DNA, which results in small fragments of the genetic material. The replication of phage DNA occurs. At the same time of packaging, the genetic material of the bacterial DNA accidentally gets into the bacteriophage by a phenomenon called false packaging. Then, the biosynthesis of bacteriophage occurs, and the phage get to assemble and finally cause lysis of the donor cell. The bacteriophage then attack the other bacterial cell, and again it will penetrate its phage DNA into the host cell. The phage DNA will incorporate in the recipient's DNA and leads to the formation of a recombinant cell. For specialized transduction, first, the bacteriophage attack the bacterial cell or donor cell. Then, the genetic material of the bacteriophage will penetrate the bacterial chromosome. The phage DNA will incorporate within the bacterial DNA, and then it will start the replication cycle. The phage DNA then control over the cell machinery of the bacteria. This causes the assembly of the bacteriophage that consists of both phage and donor's DNA. After that, a UV light induced cell lysis and results in the release of lysogen. Then, the bacteriophage attack the other bacterial cell and the cycle will continue. The new bacteria will contain three genetic components which came from the virus, that all bacteria and the host itself. So this is an example for the bacteriophages cycles that includes generalized transduction in lytic cycle and specialized transduction in lysogenic cycle. It is the same process that keep repeating to produce more and more phage. That's all for transduction.
Assalamualaikum, my name is Nur Izzati binti Nordin. I am your third presenter. Today, I would like to present about conjugation and plasmid. Number 1, conjugation is the process where genetic material is transferred between bacterial cells. Number 2, sexpili allows cells to connect and pull together for DNA transfer. Number 3, a piece of DNA called the effector is required for the production of sexpili. And number 4, the effector can exist as a separate plasmid or as DNA within the bacterial chromosome. There have several parts that involved in this process. Number 1 is villus, number 2 plasmid, and number 3 chromosomal DNA of donor and recipient. The effector as a plasmid. Cell containing the F plasmid function as DNA donor during conjugation. Cell without the effector function as DNA recipient during conjugation. The effector is transferable during conjugation. Conjugation and transfer of NF plasmid. Number 1, donor cell attached to the recipient cell with its villus. The villus draw the cell together. Number 2, the cell contact each other. Number 3, one strand of plasmid DNA transferred to the recipient. Number 4, the recipient synthesizes a complementary strand to become NF plus cell. The donor synthesizes a complementary strand, restoring its complete plasmid and become 2F plus cell. Conjugation and transfer of part of Nhfr bacterial chromosome. Number 1, the whole ring of bacterial chromosome and F factor is replicated. Number 2, the ring from donor cell to the recipient cell is being transferred. Number 3, the A plus will change the place with A minus. Number 4, recombinant F minus bacterium is formed. The F factor in the chromosome. A cell with the F factor built into its chromosome functions as a donor during conjugation. The recipient becomes a recombinant bacterium with DNA from 2 different cells. It is assumed that horizontal gene transfer is also important in archaea. The process Nhfr cell become F prime cell. In this process, crossing over will take the place within the Nhfr chromosome to separate F plasmid and bacterial chromosome and become F prime cell. If the donor is F plus and the recipient is F minus, molecule that transfer is F plasmid and the product is F plus cell. If the donor is Nhfr and the recipient is F minus, molecule that transfer is initiating segment of F plasmid and variable quantity of chromosomal DNA, and the product is F minus with variable quantity of chromosomal DNA. If the donor is F prime and the recipient is F minus, molecule that transfer is F plus plasmid and some chromosomal genes is carried with it, and the product is F prime cell with some duplicate gene pairs, one on chromosome and one on plasmid.